One of the practicals in Biology Paper 2 is the Decay Practical, if you're doing triple. Here's everything you'll need to know for if it comes up. The purpose of the practical is to investigate how temperature affects the decay of fresh milk. So you can do this in a few ways. We could literally observe the decay of fresh milk over many days, or we can instead sort of simulate the decay of milk using lipase enzyme. I'm going to focus on the example using lipase. So how will we know when the milk has been broken down by that lipase enzyme? Well, here's the science. Milk contains a lot of lipids. Lipids look a bit like this, a kind of jellyfish with a glycerol head and fatty acid tails. If lipase comes along, it will slice this bond between the glycerol head and the fatty acid tails, releasing those fatty acids. And having more fatty acids in your solution will make it more acidic. So we just pay attention to how acidic the solution is. Let's talk about the setup. 2 test tubes needed, one for lipase enzyme and one for your fresh milk. We're going to want to see clearly when the milk goes from an alkaline solution to an acid. So what we're going to also add to our fresh milk is something that's going to ensure that our solution is an alkaline. We're trying to observe when the solution goes from being an alkali to an acid. So ensure that the starting solution is an alkali, we'll also add some sodium carbonate to the milk. This way, when doing our practical, we can see a clear endpoint when our solution changes from being an alkali into being an acid as those lipids are broken down. If we are to observe such a change, we're going to also need an indicator. So a perfect indicator for this would be cresol red. It's purple in an alkaline solution and it will change to yellow in an acidic solution. Next, remember we're testing for different temperatures, so we'll now need to take both of those test tubes and place them in a water bath so that they can incubate and reach that set temperature that we're going to test. Once we've left them for a set amount of time, at least a few minutes, we can now mix together the lipase and our milk solution. Then start your timer and observe how long it takes for the solution to change from that purple to yellow, from alkali to acid as those fats are broken down. And then bosh, all you need to do is repeat that process with a range of different temperatures. And with that, you'll be able to observe how temperature affects the rate of decay. To analyse the results, you can plot a graph of temperature and time taken for the colour change. With that, what you'd observe is a nice smiley face curve. That's because the rate of lipase activity increases up to the optimum. Past that point, that lipase enzyme denatures and the rate slows down again. Great, okay, let's make sure you've understood that. Let's remember the variables. Independent, the thing we changed was the temperature. The dependent, what we tried to affect was how long it took for the indicator to change from purple to yellow. And then the control variables are everything else that could also affect that rate of enzyme activity there. That would include the volume of all of our solutions, the lipase, the milk, and also the sodium carbonate that we used, the time that we allow them to incubate for, the same exact type of milk, the concentration of our lipase solution. Brilliant. Well, how could we improve our results? Well, repeat at every single temperature and calculate yourself a mean. I hope that helps. Save this so you can watch it again before your biology exam. If there's anything else you need a quick hand with, let me know in the comments. Best of luck on Monday.